Welcome back, friends. So we talked about the structure of the flagella. Now in this video lecture we are going to talk about that how actually the flagella are synthesized. Okay, so let me write synthesis of the flagella. Synthesis of flagella. Now the flagella can be synthesized by different proteins, as I told you, means these fly proteins. So how actually these fly proteins are synthesized? Means at that cytoplasm area, means at this cytoplasm area we have a messenger RNA, as I told you. With this messenger RNA we have a ribosomes machinery. So this is the small subunit, this is the large and this is the small subunit. With this machinery what we have the means when they are connected to each other and when the ribosome moves in this direction, so then we get what? We get the proteins. So we get different proteins. Now each proteins, we call these proteins what? We call it, these protein we call it flagellin. Flagellins. Now these flagellins proteins are actually important to make this flagella portion. Now what actually happen? These flagellin proteins are, first of all, these flagellin proteins can make fly M and means this M and S ring. So once they form this M and S ring, so once these rings are formed, after that again these machinery are connected to each other and they will make then this one portion, means this fly G proteins. After fly G protein made, so then they can make fly M and fly N, means then the rotor portion will be formed. So first of all the rotor portion will be formed. Okay, after the rotor portion what we have another portion, we call it stator portion. So this is the first portion. The second portion is synthesized, we have the stator portion, which are usually fixed. Now in this case again these ribosomes and this messenger RNA are connected to each other. Now the first thing that I forget, means for the synthesis of this basal body and to this hook, means to this basal and to hook, we have almost 10 to 30 genes needed. Okay, so we need 10 to 30 genes for the synthesis of basal and hook portion. Now after the stator are formed, so then they make what? They make the P and L ring. After the PL ring are formed, so then they make this hook cap portion. Okay, so this is the third portion. After that, this is the third portion which are synthesized. Then we have a hook portion are synthesized. So what actually happen? Hook portion are usually synthesized by this flagellin proteins. Means this one area as you are watching, we call that what? We call that export gate, or sometime we call that or filament hollow internal core. So let me write in a short form: filament hollow internal core. Okay, now this hollow internal core is actually a tube, okay, where the flagellin proteins are move at the upward direction. Flagellin proteins, a lot of flagellin proteins are formed at the cytoplasm area, and then they will be moved from that portion to this export gate. So export gate, once they reach to this surface of the outer membrane, after that a cap, as I explained it in the past video, so then a cap will be synthesized. We call it hook cap. I already write it here. A hook cap once formed, so this hook cap moves in the upward direction. So when the hook cap moves upward direction, with this movement of upward direction of the hook cap, what actually happen? So once the hook cap are formed, so when the hook cap are moved in the upward direction, so then the flagellin proteins will be attracted, and these flagellin proteins are connected with each other and they are assembled with each other and added with each other, and similarly they can make what? They can make hook portion. After that the hook cap will be removed out, and then again here the proteins are formed, means flagellin proteins are formed, and these flagellin proteins are again transported to this export gate, or we call it flagellin hollow internal core. So once they are passes from that area, so then next we have filament cap are formed. So once the filament cap are formed, so then very easily the means we can say very easily the flagellin portion will be formed, or we can say filament portion it will be formed. So this cap means this filament cap protein will be moved at the upward direction. So when they move at the upward direction, so then gradually the filament or flagellin proteins are connected and assembled with each other, and they make gradually the filament portion. Okay, so if we see, so this filament are actually synthesized to this basal, means this basal portion. Okay, so this filament are usually synthesized with this hook, or we can say this hook portion are synthesized with this basal portion. Now, as I told you that there are 10 to 30 genes which are usually involved for the formation of basal and hook portion, and the rest of the protein are actually responsible for the filament synthesis. Okay, so the rest of the protein are actually involved for the filament portion of filament synthesis, and some of them protein are actually responsible for their function as well, that where the cell can exact go. Okay, so this hook portion and this filament portion are actually self-assembly. Why? Because they have a caps protein. The first we call a hook cap and filament cap proteins. With these caps proteins, they are the self-assembly. Okay, so that's all about the synthesis of the flagella, that how actually the flagella are synthesized. I hope you understand about the flagella synthesis. Inshallah, in the next video lecture we are going to talk about that how actually the flagella can perform their function, or we can say that how actually the means that with the help of flagella how the bacteria can move from one direction to another direction. Okay, so stay tuned and continue to watch the rest of the video. Thank you so much.